Hello, everyone. Welcome to today's practice session. I hope everyone's weekend is going great. It's just going to be me today leading the session, but we're still going to do 4 passages together. All right, how did you guys find this passage? Was it easy, medium, hard? Yeah, this one was pretty challenging. All right. So what did you guys get for question 1? If you would like for me to call on you to explain your reasoning, just type an asterisk next to your answer. Yeah, so it was a little bit tricky to find the structure for compound 1, especially with the time limit. So let's go through it together. All right, the passage states that the compound has 2 degrees of unsaturation, meaning that it could have 2 double bonds, 2 rings, or a double bond and a ring. The IR data says that there is a peak at 1705 centimeters, meaning that there has to be at least one carbonyl group in the structure. The HNMR data also tells you that there is a ring in the molecule because, sorry, it does not tell you that there is a ring in the molecule because there are 3 peaks, meaning that there are at least 3 different chemical environments. So the other degree of unsaturation must be accounted for by either a double bond or another carbonyl group. The last paragraph gives us a hint that we can use. When we treat compound 1 with NaBH4, the degrees of unsaturation is 0, meaning that the carbonyl groups must have disappeared. So if we treat compound 1 with NaBH4, we are hydrating the carbonyl groups so that they become 2 alcohol groups. So I drew what the reaction would look like at the bottom. We can also see that there are 3 groups of hydrogens, all in different environments, which accounts for the 3 signals that we see in the NMR spectrum. So there isn't like a clear-cut way of figuring out a structure. It's really just trial and error. You really have to like use all of the clues that the passage gives you. So like 3 signals, 3 chemical environments. You have like the neighbors here, the splitting pattern, and you also have the IR spectrum that's telling you that there's a carbonyl group. And also using the hint that like hydrating with NaBH4 gives you the alcohol groups. Does that make sense? Yeah. Well, I'm not entirely sure about 2950. I know that like in the 3300 is the alcohol group. I think they just threw that in there to like confuse you.
I was stuck on figuring the structure because I thought the first IR value is very close to 3000, which is supposed to be alcohol, and the second one is a ketone.
So the alcohol actually absorbs at 3300 or like 3350. So it's a bit far off.
But if you have 2 different values, how could you know both of them are ketone?
So the ketone is represented by this value at around 1700.
And what's the
So
first value suggest?
I'm sorry?
What is the 2950 suggest?
Honestly, I don't know. Like for the MCAT, you have to know like the 1700 and the 3300. Like those are the 2 most important things that you have to know.
Okay, thank you.
Yeah, so sometimes the MCAT will give you like random pieces of data that you don't really like need. Like the 2950 doesn't really tell you anything. Oh, so degrees of unsaturation. So the passage told us that it has 2 degrees of unsaturation. So there's a formula that you have to have memorized. And basically the degrees of unsaturation tells you how many double bonds there are, or if the structure has like a ring or a double bond and a ring, etc. So in this passage, they gave us 2 degrees of unsaturation, which means we have 2 double bonds here. Is everyone clear on that?
Yeah, that made sense. But I was just wondering how you knew that it wasn't corresponding to a ring.
So because there were 3 signals in the NMR data, so in a ring, you kind of have everything like symmetrical, sort of. You would have fewer signals because everyone would kind of be like a neighbor, if that makes sense. So you could like try it out with a ring, but you like that you wouldn't get the same number of signals and the same splitting pattern.
Okay, so the 2 degrees of unsaturation, it could have been a ring, but because of the pattern of the, I guess the splitting, that's how we knew it wasn't a ring.
Yeah, and also looking at the molecular formula, they gave you 2 oxygens. So it would make sense that the degrees of unsaturation would come from a ketone rather than a ring.
Okay, and sorry to just ask, I just wanted to follow up with the splitting. What about that showed us that it wasn't a ring? Like, what are we looking for in the singlet, triplet, and quintet?
Yeah, so for the first one, singlet, it's telling us that 6 hydrogens do not have a neighbor. So if we look at the structure, we have 1, 2, 3, 4, 5, 6 hydrogens that are in the same environment, but they don't have any neighbors. So that corresponds to the structure. So for the 4 hydrogens, they have a triplet, meaning that 4 hydrogens have 2 neighbors. So if we look at the structure again, we have 1, 2, 3, 4 hydrogens in yellow. These hydrogens are in the same chemical environment because they have the same neighbors, and they have 2 neighbors, which are these 2 hydrogens here. So that corresponds to this. And then 2 hydrogens split into a quintet, which means that they have 4 neighbors. So looking at the structure again, we have these 2 hydrogens in blue, and they also have 4 neighbors here. Does that make sense? Okay, great. All right, does anyone have any more questions before we go to question 2? Okay. All right, what did you guys get for this question? All right, so the answer is D because, like I said before, one of the values for IR absorption that you have to have memorized is 3300 or 3350, which represents an alcohol group. So this question is purely like recall. Does anyone have any questions? All right. And what did you guys get for this question?
For the previous question, is carboxylic acid also close to that range? Are they overlap in value?
So carboxylic acid would have a different value. I don't know off the top of my head what that value is, but it would be much different than alcohol because it has that, like the double bond O and then the OH.
Because I think the carboxylic acid group is 3,300 to 2,500.
All right, let me see. Yeah, you are right. But for compound 2 specifically, what they're asking about is you would have to choose between a carboxylic acid and an alcohol. And the passage told you that we're treating compound 1 with NaBH4. And what NaBH4 does is that it hydrates the ketones that I showed you before into alcohol groups. So it doesn't add an oxygen. Does that make sense? I can show you here. So when we add NaBH4, we get these alcohol groups. We're not getting a carboxylic acid. Yeah, it's based on the previous question. Okay. And what about this question? What did you guys get? Okay, Brian, why did you get A?
Because according to the formula, you would multiply the number of carbon and then minus the number of hydrogen and then divide everything by 2. And oxygen is unaffected by the unaffected to the unsaturated number. So you will get one unsaturation in the end.
So you missed like one little tiny detail in the equation. So to find the degrees of unsaturation, you have to multiply the number of carbons by 2 and then add 2 and then subtract the number of hydrogens and then divide by 2. So we would get 2 degrees of unsaturation.
Oh, I see. Thank you.
Mhm. Great. And what did you guys get for this question? Yeah, let me do that. All right. So I'm gonna accidentally show you the next slide because I can't go forward unless I go back. Tell me when you're finished with this slide. All right. And what did you guys get for this question? Okay, let's go over it. So the correct answer for this question is D. So as we know, the pheromone has a signal at 1705, which means it has a ketone. Treating a ketone with a Grignard reagent gives us a carbon-carbon bond and an alcohol, as we can see in the reaction below. And that alcohol will absorb at 3350 on the IR spectrum. Does that make sense?
Is it suppose different because I look up on the table and they have different value. I look up on the mile review sheet and they say it's 3,000 to 3,300. If the number is very close, what should we do?
So the way I memorize it, I like even in my orgo classes, I memorized it as like 3,300-ish. But for MCAT purposes, I would go to the reviews. So do you know like the content breakdown the double AMC has?
Yes, I know the outline.
Yeah, the outline. So I think somewhere there it would tell you, but also you could look at the double AMC like full lengths and the chemistry like questions. Like I'm sure it has that. And I even saw on the double AMC question bank that that they want you to recognize that 3,300 is the alcohol group.
I see. And because I think usually if the number is very close, they will usually, in my orgo class, they will usually tell you if there's a broad signal or narrow signal, and then you would determine it from that.
Yeah, exactly. How do you know what products are formed from this? So to answer lots of orgo questions, you would have to memorize the reagent and like what they create. So the Grignard reagent adds that carbon-carbon bond here, and also it hydrates the ketone into an alcohol group. Does that make sense? You don't need to know like the exact mechanism. That one's like beyond the MCAT. So the Grignard reagent, you know how this, so a Grignard reagent is basically like any carbon group. So it could be CH3, it could be like any carbon skeleton attached to MgBr. Yeah, and then so it basically just drops this carbon group into the carbon that's attached to a ketone. And while it does that, it creates this carbon-carbon bond, and also it hydrates the ketone into an alcohol. I actually do in my notebook, so I can make it, I guess, readable and then send it to you guys later. All right. And what did you guys get for this question? Yeah. So for this question specifically, compound 2, as we know, has two alcohol groups. We deciphered its structure earlier, and now all we have to do is count how many different hydrogen environments there are to get the number of signals in the IR spectrum. So if we go back. Sorry, I'm looking for it. All right, if we go back, we can count how many different hydrogen environments there are. So due to symmetry, there are only 5 signals, and you can see like an internal line of symmetry here. Does that make sense? Okay. All right, that was the last question. Do you guys have any questions about this passage before we go to the next one? Yeah. So for this question, like I said, you have to have memorized your reagents and what they do. So the Grignard reagent is, why 3,500 versus 3,100? So it's 3,350 because, so the alcohol group, the double AMC wants you to recognize that a peak at 3,350 corresponds to the alcohol group. 3,100 is too low for it to be considered an alcohol, but that's the main distinction. Mhm. All right, and how did you guys find this passage? Medium, okay. So what did you guys get for question 1? Okay, Brian, why do you say C?
I feel this question is kind of like a random fact question that you would just need to know the yeast is eukaryote and this unicellular. And because I think yeast is like a kind of similar to fungus, so most characteristic of the fungus has the spore. So that's why I pick C.
Yeah, so yeast and mold are both fungi, so they're both eukaryotes, so A is out. Fungi remain haploid for most of their life cycle, and they also produce spores. So yeast and mold have that in common. Yeast are actually unicellular, whereas mold is actually multicellular. So that's why B is our answer. You're right that this is kind of like a random fact. Does that make sense?
I thought yeast is unicellular.
So mold is actually multicellular. It's just like, it's kind of annoying that you have to know that, but it is. What helped me recognize or remember these random facts is, do you know the website Varsity Tutors?
No.
Okay, so if you Google like MCAT biology fungi questions, Varsity Tutors should be like the first one that pops up. And this one has like a lot of random facts that they quiz you on, and that kind of helped me, I guess, remember some of these.
Okay, thank you.
No problem. All right, and what did you guys get for this question? Okay, so the correct answer is C. So the passage stated multiple times that the cells are undergoing mitosis and crossing over meiotic division and reduction division are all characteristics of meiosis, so they have nothing to do with mitosis. So once the mutant cell reaches the end of mitosis, it's unable to undergo a new round of mitosis. So therefore, it's not able to reinitiate DNA duplication. Does that make sense? So D, reduction division is a step in meiosis 1. So it's just when the chromosome number drops from 46 diploid to 23 chromosomes haploid. So that doesn't happen in mitosis. So like the daughter cell is identical to the parent cell in mitosis. Does that make sense? Yeah, okay. All right, and what did you guys get for this question? Okay, so for this question, choice A was incorrect because the behavior of the wild type cells, which I underlined, doesn't tell us anything about the block in the cell cycle in the mutants. So the question is asking us about the mutants specifically. Choice B is wrong because it's telling us that at a certain temperature, the mutants are able to enter mitosis, but it doesn't necessarily tell us anything about the specific stage in which the cell cycle is blocked. And choice D is wrong because it's a bit unclear as to how the addition of regulatory proteins can allow for the mutant cells to grow. And it doesn't tell us anything about the specific stage. So choice C has to be our answer. If all of the mutant cells were blocked at the same point in the cell cycle, then there must be something wrong with that specific stage of the cell cycle. So the fact that the experimenter placed the cells at high temperatures at different times tells us that there has to be a defect in the cell cycle that isn't letting the cell pass through. So the key to answering this question is to think about each option and ask yourself, how is it relevant to the question and to what each option implies? So choice C is the most relevant to the question because we're talking about mutants and it talks about the specific point in the cell cycle. And it implies that regardless of time exposed to high temperatures, the mutants were halted at a specific point in the cell cycle, meaning that time isn't a factor, so it must be the mutation of the CDC gene that is causing the halt. Does that make sense?
But if you're stating the wild type is able to pass their cell, and would it kind of imply that this one, the wild type has an advantage over the mutant cell?
Yeah, but it's telling us about the wild type cells, but what we're focusing on is the mutation of the CDC gene. So we're looking for something that is telling us about the behavior of the mutant cell.
I see. Thank you. It's more direct.
Yeah. Does anyone have any more questions about number 3? Okay. So what did you guys get for this question? Yeah, Brian, why did you get D?
Because the competitive advantage is basically talking about the ability of adopting the changing environment and then pass down their trait. And if they say the wild type have advantage over the mutant type, then the wild type must be able to like a kind of outpass in their offspring in
Yeah,
terms of the
exactly.
number of population.
Yeah, you're exactly right. So the correct answer is D. So like you said, the question is asking us which of the following is evidence that wild type yeast have a competitive advantage over the CDC mutants. So like Brian said, choice D is saying that in a plate with both wild type and mutant yeast, only wild type yeast are found on the plate after 24 hours, meaning that over time the yeast predominates. So this option answers the question directly, which makes it the best answer choice. Option A is irrelevant. It's unclear how a spindle apparatus forming during late interphase would prove to be a competitive advantage. Option B is also incorrect. It doesn't explain how wild type yeast have a competitive advantage again. And finally, although option C is telling us that the mutant cannot grow at high temperatures, which would probably lead to a competitive disadvantage, option D is still the better choice because it answers the question of competition directly. Does that make sense? Okay, I'll take that as a yes. And what did you guys get for question 5? Okay, so to answer this question, it would be helpful to have the cell cycle memorized, or at least knowing that the cell synthesizes enough material to divide into 2 separate cells. So the genome replicates throughout all of interphase. So if the cell is unable to enter interphase, then there's no way that the genome can replicate. So there is no evidence that A, C, and D could be correct. A especially is incorrect because although DNA replication can stop, the cell could still synthesize proteins. C is incorrect because the parent cells have to be haploid if the daughter cells emerged as haploid. So there's no way that a diploid cell can undergo mitosis and divide into a haploid cell because the passage states that the daughter cells were identical to the parent. And D is also incorrect because there's no indication that this could occur in the question stem. All right, and last question. What did you guys get for this? All right, Brian, why do you say A?
Because if the mutant cell is recessive and they have the wild type is like assumed to be all dominant of the wild type, then there will just be homozygous of the wild type multiplied by the homozygous of the recessive.
Mm-hmm. Yeah, you're correct that the answer is A. So the question stem is telling you that the CDC mutation is recessive. So the wild type phenotype, the wild type phenotype will dominate. So based on this, we can eliminate C and D. And because we are fusing 2 different genotypes, the mutant and the wild type, the genotype will be heterozygous. Right. Does anyone have any questions about the last 2 passages?
I have a question on the IR value.
Yeah.
So you say the exact number could be found in AANC website?
So
or like in the question bank?
So the way that I found the exact numbers is, I mean, like I have the TPR textbooks, which told me like 3350 or like 3300 corresponds to OH, but like I understand like a lot of people have different values for them. But if you do like the question banks on AAMC, they would tell you like 3300 or 3350 corresponds to OH. So did you buy like the online bundle? Because if so, then
yes,
one of those questions will tell you. Like
yes, I buy the online bundle.
Okay.
I also have a Kaplan and TPR. Does the value being accurate?
So for TPR, I know they're accurate. For Kaplan, I'm not sure because I haven't read them.
Okay, so I could just use the one in the TPR.
Yeah.
Okay.
I did not create these passages, and they are not from the double AMC. They are actually from the Princeton Review Science Workbook. So if a question is too difficult or too easy, I will modify them. But so far, I haven't modified it. I haven't modified these questions because they're sort of what you can expect.
Yes, thank you.
Mhm. All right, so I'm going to send you the links to the next two passages that we're going to do together. I'll give you a maximum of 20 minutes to complete them, and once you're done, you can type done in the chat. All right, we'll go ahead and get started. So how did you guys find this passage? Was it easy, medium, hard? Medium, okay. All right, so what did you guys get for this question? Again, if you want me to call on you, you can just type an asterisk next to your answer. All right, so for this question, the correct answer is D. So only the nucleus of the cell that was arrested in G1 was transplanted, which was later activated after the transplant. Therefore, the substance that was in control of G1 arrest was not the nucleus or inside of the nucleus, but has to be outside of the nucleus, which is the cytoplasm. So A is wrong because if the nucleus was in control of G1 arrest, then the transplanted nucleus would still be in G1 arrest. B is a little vague, so someplace within the cell could be the nucleus, it could be the cytoplasm, but we don't know from this one sentence. And that's why D is the better answer. So C is wrong. It's not specified in the experiment if we're dealing with plant cells, since animal cells don't have cell walls. Yeah, does that make sense?
Can you please explain why A is wrong?
So for this experiment, we're transplanting the nucleus here that's in G1 arrest, and we're transplanting it into this cell that's in S phase. So once we do that, we have this empty cell, and the transplanted nucleus is finally able to go into the S phase. So because the transplanted cell is no longer in G1 arrest, we can conclude that the nucleus is not responsible for G1 arrest, because if it were, then the transplanted cell would not be in S phase. It would be in G1 arrest still. So that's why we can, yeah.
Thank you.
All right, and what did you guys get for this question? Yeah. All right, Brian, why did you get C?
Because I remember the typical illustration on the textbook that is showing the cell will first condense, then when the membrane break down, you will immediately see the condensing chromosome.
Yeah, exactly. So in general, the first sign of mitosis that is visible is a condensation of chromosomes during prophase. So that's why C is our answer. Yeah, the formation of nuclear membranes occurs during telophase. The disappearance of the nuclear membrane occurs during late prophase, and the splitting of the chromosomes occurs during early anaphase. So yeah, you can remember the acronym PMAT. So the first stage is prophase, next is metaphase, next is anaphase, and then telophase. All right. And what did you guys get for this question? Brian, how did you get A?
I do the by process elimination because the mature red cell will not have the nucleus. Therefore, it's unlikely they will do any kind of division, and that's why I eliminate B, C, D.
Yeah. So item 1 is wrong because primary spermatocytes undergo meiosis to divide into secondary spermatocytes. So we can eliminate B and D. I included a diagram to help you visualize spermatogenesis. I know it's kind of like a complicated diagram. But yeah, so only premature spermatogonium divide into primary spermatocytes, which then divide into secondary spermatocytes via meiosis 1. So item 3 is wrong because, like Brian said, mature erythrocytes are terminally differentiated and lack a nucleus in humans. So it's impossible for them to undergo mitosis. Yeah. Does that make sense? Okay. So what did you guys get for this question? Yeah, so for this question, the correct answer is D. So in both mitosis and meiosis, chromosomes replicate before beginning cell division with prophase. So option A is wrong. Crossing over only occurs in meiosis in order to get diversity. So daughter cells have a combination of chromosomes that are passed on from both mom and dad. In mitosis, the daughter cells are identical to the parent cell. Finally, option C is wrong because in mitosis, there is only division to make 2 daughter cells, but meiosis has, so there's only 1 division to make 2 daughter cells, but meiosis has 2 divisions to make 4 daughter cells. Okay. And what did you guys get for this question? Okay. Brian, how did you get A?
I think the first growth phase is more prepared for the cell membrane and other organelle, and the S is synthesis in their genome.
Yeah, so for this question, the correct answer is B. In the passage, we're told that in G1 phase, organelles are replicated, so like the mitochondria. A is incorrect because during the S phase, cellular chromosomal material are replicated. So it could be tempting to pick A because the passage says that chromosomal material is replicated, and the question is asking about the genomes of mitochondria and chloroplasts. But the distinction is that in the S phase, only the cell's chromosomes in the nucleus are replicated. And since the mitochondria is an organelle, it will duplicate in G1. C is wrong because organelles are not synthesized during mitosis. This occurs before mitosis, so like during interphase. And D is wrong because cytokinesis is the splitting of 2 cells. So yeah. Does that make sense?
So they will be based on the organelle, not based on if they are genome.
Right.
Okay. I got it. Thank you.
Okay. And last question, what did you get? Yeah, this question is pretty much recall. So like I said, you can use the mnemonic PMAT. So prophase, metaphase, anaphase, telophase. That's the order of the stages of mitosis. Right. Does anyone have any questions about this passage before we move on to sociology? I know that there's a minute left, so it's okay if you guys have to go because this session is being recorded anyway. All right, how did you find the passage? Was it easy? I hope. Yeah. Okay, let's go over it. So what did you guys get for question 1? All right, so let's start with item 1. So item 1 is true. Both groups of children aged 0 to 5 have a higher life expectancy than the 2 groups of children aged 6 to 10. So we can eliminate B and D. Now we just have to see if item 2 is true. So item 2 says that children whose parents have full-time employment will have a higher life expectancy than children whose parents have contingent employment. In figure 1, we can see that the black column is always taller than the gray column. So item 2 is true and C is our answer. Item 3 is wrong because while it is true that the younger children have a higher life expectancy than the older children, within each age group, those who have parents who are employed full-time have a higher life expectancy. So the type of employment is still a better predictor than age. Does that make sense? Okay. And what did you guys get for question 2? Okay, Brian, how did you get C?
Because the intersectionality is basically talking about the multiple identity and how they interact.
Yeah, exactly. So this question, to answer this question, we have to know our definitions. So like Brian said, intersectionality is the view that the lived experiences of individuals cannot be broken down to only one of their identities. Instead, someone's lived experiences should be viewed as an intersection of multiple identities. So in this question, we're told that the African-American woman's identity must take into consideration her race and her gender together, not as two separate things. For example, a white woman and an African-American woman can both identify as a woman, but they don't necessarily have the same experiences because of their race. So option A is wrong because symbolic interactionism is a theory that states that people act towards things based on the meaning that they have towards the references. For example, a stick figure with a dress means woman because we associate dresses with women. Option B is wrong. Social identity theory defines the way that individuals shift their social and personal identities based on who they are talking to. For example, the way I interact with my boss is very different than the way I interact with my friends. And finally, option D is wrong because social constructionism is the theory that people develop their knowledge of the world in a social context. So basically, things do not have stable meanings, but they gain their meanings from people who use them. An example is money. Money has so much value because we give it value. Does that make sense? Okay. And question 3, what did you guys get? So in this passage, we're told that the Haitians are very close to their family members and live in a tight-knit community, meaning that they have high social support. Social support includes advice, emotional support, or financial support. However, later in the passage, we're told that the Haitians have a difficult time finding full-time employment, so this would decrease their social capital. The Haitians are unable to join social networks to gain economic capital, so that's why they have social capital. And that's why A is the correct answer. Does that make sense? So they have a tight-knit community amongst each other because they're living in like a Haitian neighborhood and they're very close to like their other Haitian neighbors and their family members. So that's why they have this tight-knit, like this tight-knit community gives them high social support.
So how do you know? So the residency giving them cold shoulder is not consideration?
Well, it does, because if there was just like one Haitian family living in a community that gives them the cold shoulder, then they would have low social support. But because there's multiple Haitian families living in a neighborhood together, they all hang out together, they all support each other because they're all Haitian. So they have high social support amongst each other.
I see. Thank you.
Okay. All right. And for this question, what did you guys get? Yeah. So for this question, we have to know our fathers of sociology. So Karl Marx was a sociologist who focused on economic capital, and he's like the father of conflict theory. He proposed that every society is divided into two classes, right? The bourgeoisie owns the means of production, and the proletariat or the working class provided labor. So this division would then create inequality, which is the heart of conflict theory. Emile Durkheim is the co-founder of structural functionalism along with Herbert Spencer. The theory states that a society is composed of systems like the human body, which all must work together to keep the system functioning. George Mead's work was important for symbolic interactionism, which views society as composed from everyday interactions. And Finally, while Max Weber did contribute to conflict theory, he critiqued Weber's theories and stated that they were too extreme. Weber did not believe that the collapse of capitalism was inevitable. He said that several factors such as agreement with authority figures and such helped maintain capitalism. Does that make sense? Okay. And what did you guys get for question 5? Yeah, so the correct answer is B. A decline in employment rates is a sign of social exclusion, not an increase in employment rates. So crime rates are correlated with social exclusion because if people can't get formal employment, they'll transition to informal quote unquote employment, like theft or prostitution. So A is wrong. C is also wrong because substance abuse rates are likely to go up with social exclusion as a method to cope. And finally, D is wrong because as the Haitians are socially excluded, a greater percentage of the population will be forced to give up their culture and their values so that they are no longer excluded. So this would be forced assimilation because the Haitians otherwise have no choice of social mobility or inclusion. Right. And last question, what did you guys get? Yeah, Brian, how did you get C?
Because they will focus on the children of the immigrants. So, and basically the only thing that will focus intergenerational relationship in C.
Yeah, exactly. So the first sentence of the passage states that the researchers are trying to study the effect of social stratification on health and well-being. So one thing about social stratification is that it can carry over from one generation to the next generation. So if someone's parents are disadvantaged, the children are also more likely to be disadvantaged. A is wrong because studying the effect of the health of other Olympians doesn't really have much to do with social stratification. Option B is also wrong because the researchers are studying social stratification, not the children's coping mechanisms. And D is wrong because the passage doesn't really state that the whites will exclude the Haitian children. In fact, since the Haitian children were born in America, they are more assimilated and less likely to be excluded. So C is our answer because it addresses the researchers' aim of studying social stratification. All right, does that make sense? Okay. So that is all for today. Thank you guys for coming to today's session. We'll be sending you the newsletter later, so make sure to sign up for next week's sessions. Oh, thank you. All right, thank you guys for coming on a weekend, especially.